Hi, I'm Lea from the University of Washington and CMAP. And obviously, all of our endeavors have been greatly affected by COVID-19. For everybody, it put the brakes on our new lab work, but others were able to put their tech dev skills to work in the fight against COVID-19. This includes our host, Jennifer Doudna, and the many others who stood up testing labs, and still more who are working on developing new and improved testing technologies. And today, I'm going to tell you about my side hustle with the Seattle flu study and our experience identifying community transmission in the United States and developing new protocols to get around supply chain problems. So the Seattle flu study was launched in the fall of 2018 with the lofty mission of transforming how infectious disease outbreaks are detected, monitored, and controlled. Since there was not a pandemic in 2018, the idea was to use the emergence and spread of the seasonal flu as a model, hence the name. The vision required lots of innovations from participant recruitment and community buy-in to get all the samples we needed, high throughput testing and sequencing, and a flexible data warehouse, and obviously the bioinformatics and modeling teams. For sample collection, if we're going to deeply sample a population, first you have to get the samples. The part of this project was led by infectious disease specialist Helen Chu for adults and Jan England for children. And the first way we got the samples was the easiest. This is the equivalent of medical dumpster diving, taking remnant samples or leftover samples from hospitals. But we also wanted people who were not seeking medical care, right? Medically attended respiratory disease is the tip of the iceberg. We wanted people who were not that sick, just to see, how it was spreading in the communities. So we had pop-up kiosks in the city in heavily trafficked areas, and the recruitment avenue that would become most important later on is our swab and send program, where folks experiencing flu-like symptoms could hit our website and sign up for a kit that we would send to their house. They would swab their own nose and send it back to the lab. All right, so with all these samples coming in back to the lab for processing, we needed more firepower. So we called in the pros, and another big name in the NHGRI in genomics was Debbie Nickerson and her Northwest Genomics Center. They had the automation and LIMS for a project of this size, and we would end up dealing with 23,000 specimens just in the first couple of months. First step in our detection pipeline is extraction, and we had an automated extractor. This thing could crank out 96 samples every hour. And then we had to find out which respiratory pathogens were in each of these samples, and we used Thermo's open array platform. The TACMAN assays are arrayed in this chip. Each through hole contains a single or multiple probe sets. Each chip can hold 48 samples, and our design could test for 27 pathogens in duplicate. The machine holds 4 chips, so we can run nearly 200 samples at a time. I realize that this is caveman technology for this crowd, akin to banging rocks against other rocks, but RT-PCR is a classic for a reason. It can accurately detect down to just a few molecules. So when we identified a pathogen that we were interested in, we moved it onto sequencing in support of the genomic epidemiology by Trevor Bedford's group. This is a screen grab from nextstrain.org, and you can see all of the flu genomes for H3N2 on the left and flu B Victoria on the right. The red ones are from the Seattle flu study, and we were producing 15% of the world's genomes before we stopped. First, from the dates on the bottom, you can see that there was a ton of H3N2 in 2018-19, but almost none in 2020 or 2019-2020. And you can see the opposite is true for flu B, with almost none in 2018 and a lot running up to flu season in 2020. You can also see there are many introductions into Seattle throughout a season. And what you can see for H3N2 is that this non-vaccine match introductions are the ones that can take off and form big transmission chains, but you can see the same, you can see in flu B that there are many introductions that manage to create all of their own little mini epidemics. And it turns out that this is how epidemics start and spread, lots of embers that eventually ignite. You'll also notice that these phylogenetic trees end in about mid-January, and that's when we stop paying so much attention to the flu. So about January 21st, I'm at dinner with Trevor Bedford, and he says, from the genomic data, which you can see here, there's not a whole heck of a lot of variation in these very early viruses out of China, and this is actually Thailand. It's obvious that this is spreading from human to human. And he says, from what we know, this could be as bad as 2018. Sometimes going to dinner with an epidemiologist is kind of a bummer, but anyway, we've built this respiratory surveillance platform, so of course we are going to use it. Now we need to develop a test, and the CDC's version had just been published, and I nearly fell off my chair. It was so low throughput, 22 samples at a time. You see this huge pandemic coming, and the CDC designs a test that does 22 samples at a time. So we developed our own multiplex laboratory developed test, and we were ready to go at 190 samples at a time. And then we waited. Nobody could give us a straight answer if we were allowed to start testing specimens from our amazing trove that we're running through my lab. For example, we were told by the governor to spare no expense, and then his Department of Health told us that we were research and not CLIA, and they wouldn't want to know if we had found one. Anywho, it was getting more obvious to our modeling team that COVID was out there, so we decided that a reasonable person would run the samples anyway, and we would figure out what we would do with a positive if we found one. Turns out that that was 2 days later. So of course we sequenced the genome as fast as we can. So as Trevor points out in this tweet, because this is how science was distributed back in February, the sequence has enormous consequences. It looked at the time like it was related to a travel case or Washington 1, which was the original case from 6 weeks before, and that our modeling suggested we had a pretty sizable outbreak in our own backyard. So fast forward, so we'll stop here. So we've discovered community spread, and we think, and an outbreak right in Seattle. And now we're going to actually fast forward to October. So besides the unabating pandemic in the United States that has killed 200,000 people, it's Groundhog Day out here for COVID testing labs. With no coordinated United States response, we're still having massive supply chain disruptions. So we've been focused on removing as many steps from the protocol as possible. First, we got rid of the fancy swabs and transport media. And hopefully by the time you're hearing this talk, that we'll have presented a validated nasal swab extraction-free protocol to the Department of Health. Sanjay, who did this work and will be here to answer questions, has shown that it is nearly as sensitive as the standard protocol, and obviously way faster and cheaper. I could go on, and I'm especially sad that there is no cocktail hour because, hoo boy, are there stories. But I'd like to thank you all for listening and thank an amazing team for all of the work that was put into the Seattle flu study. This is just the molecular team and the PIs, by the way. And hopefully we all get back to genomic technology development sometime soon. Thank you.